During this week, I will explain you, well, I will give you 3 classes, okay? All of them have the same, more or less the same subject, and I will try not to be repetitive, but some slides will be the same, just to look it from another focus. Today, I will give you an introduction about what is regeneration, a little bit of embryonic development, for you to see the importance of the embryonic development in all the things that we are studying that are regeneration and the therapies with cells, okay? So the class of today is a general introduction of the concept of cellular therapies, stem cells, embryology, and regeneration. And I will talk to you about the mechanism of regeneration in the animal kingdom. Okay, not in humans, but in animals in general. You already had this slide. We know that cellular and tissue therapy refers to the process of introducing cells or tissues in one organism with the aim to treat illness. And for do that, okay, regenerative medicine is the field of the medicine that studies this process, and you know that this is a multidisciplinary science. Many experts of many fields must be involved. For example, developmental biology, okay, it's essential because we cannot talk about the stem cells without knowing the process of development, of normal development. Embryology, okay, and also regeneration, because during regeneration we need the stem cells, the body needs the stem cells or any other kind of cell to substitute the missing tissues. So we must know about all these processes. And a part of that, we must know about how to introduce these cells in the organism. We need engineering. We are not going to talk about it, but we need it. Now we already know that we have 2 ways to introduce cells to treat illness. The first one is the rapid isolation and reamplification of cells. For that, we don't need so much knowledge, okay, because we don't need to transform or to transform the cells. But the second one is the more promising one. It's the cell isolation, transport to the lab, and extensive in vitro manipulation before the reimplantation. And this is what gives us the new opportunities to treat illness. For that aim, we have these 2 main classes of cells, ES cells and EPS cells, and we will talk about them later on. Okay, so why do we need to know about developmental biology and embryology? Because it's in the process of development that from an egg, okay, from a zygote that is fertilized, many cells must differentiate to the proper fate and must organize themselves. In this process, for example, the different germ layers, ectoderm, mesoderm, and endoderm, are differentiated in a natural manner. But we need to know how animals do it. If we want to treat humans, maybe we need to know how humans do it. Then to be able in vitro to take a cell and induce them to differentiate, for example, to an endodermal fate, to a gut, for example, or to an intestine. We need to know first how to get the endodermal fate. Okay, this is maybe the first decision. And to know these things, we must study the embryogenesis of humans and other animals, because humans are not so available, it's not so easy. So many times what researchers do is to study the development of other animals, okay? Animals that are established as a model in the lab. Okay, have any one of you studied embryogenesis? Any subject about embryogenesis and embryonic development? Yes or not? See, 2? Only 2? For me this is important because embryonic development is not an easy thing. So if you have never studied it, then I must take in trouble. Okay. What is embryogenesis? Embryogenesis is the development of an embryo from a fertilized egg. Okay? Developmental biology is the science that studies how genes control embryonic development. And what is the problem? What is the question that wants to be solved? Is how from one cell we can reach so many cells so well organized, okay, in a space and in time. How can it happen? And then, compared with that, because most of the things that happen during development, during embryonic development, must also happen during regeneration, we must study what is regeneration. And regeneration, it's not the generation, the new generation, okay, of things from nothing. It's the proliferation of cells that give rise to the missing tissue that was already there. And this is different from an embryo, because an embryo, it was not there, but during regeneration you must reestablish something that was there before, okay? And the problem or the question that we want to solve with regeneration is where the cells come from, okay? Where do the cells come from? If, for example, if I cannot regenerate a limb or an arm, but salamanders, for example, can do it, okay? Where the cells come from to regenerate a new limb? And how do they do to make again the correct number of digits, for example? How they do it? So the signals that are used during development, embryonic development, and during regeneration are very similar. Yeah? So, you know, now the main problem we want to study during development is how from an egg, okay, that it's fertilized, it's only one cell, it starts to divide and gives a so well-patterned organism. Here is an example of a model that is very well known in the lab. It's Xenopus. And in Xenopus, just for you to have an idea of what is embryogenesis, first we have at the stage 1, we have the egg in which an spermatozoid fertilized, okay? And then it comes the stage of the cleavage. The cleavage just means that the cell begins to divide a lot. These divisions are very rapid, okay, and if you look at the size, it's not exactly like that, but normally the egg is very big, and what it does is to cleave in smaller cells. Then from what is called the blastula, that is this big ball of cells, something very, very important happens. Some people say that this is the process, the main important process that happens in your life. That is gastrulation. Okay, because during gastrulation, you go from a ball of cells to organisms that already have all the germ layers. And this is the process of gastrulation. Because from one side of the blastula, it begins to migrate the cells inside, okay, to begin to migrate to form the germ layers. Now the ectoderm, the endoderm, okay, and the mesoderm. And later on, it will come the organogenesis, that is the formation of the organs. Now that we have already the 3 main germ layers, then we will be able to form the central nervous system, for example, with the brain and the nerve cords, or the hair, or whatever, okay, the organogenesis. In the case of Xenopus, we also have a stage of metamorphosis, because the development gives rise to a tadpole, and it must transform through a metamorphosis to a frog. But it depends on the animal. This is the main stages of any developmental process, but many, many different things happen depending on the animal we are studying. For example, this is zebrafish, that it's a model also very used in the lab nowadays. And the development is very, very similar to the one of Xenopus, but this is not surprising because they are phylogenetically very close one to the other. Okay, the stage of cleavage. The cleavage is in the top of this mass of yolk. Okay, this is the yolk of the embryo, and the cleavage proceeds, and this is the gastrulation. You see the ingression of the cells to form the layers, and then the organogenesis. And again, you have a free-swimming hatchling, okay, it's like a larvae that transforms to the adult. In this case, the development is very different. This is a Drosophila, okay, but the stages are very are comparable. Okay, from Drosophila, it becomes they lay an egg that is transformed to an embryo, and inside it transforms to a different kind of larvae, and at the end to a pupa that through metamorphosis transforms to a fly again. Here in the Drosophila, there are some organs or some tissues that are very well known for scientists in the lab. This is maybe the most common or the most used model in the lab. And one of the things that the scientists study very much are what is called the imaginal disc. Do you know what are the imaginal discs? Somebody of you know? No? Digamos que no. Y tú es que también lo hicieras. Okay. Imaginal disc that at this stage, okay, of the larva, the larvae does not seem a fly. Okay, it has the shape of a worm, okay, but not a fly. And what you can see is that some cells are organized, okay, in a specific areas, and these cells are called the imaginal disc because you have, for example, two kinds of this mass of cells that are called the imaginal disc of the antenna and the eyes that in the adult will give rise to the antenna and the eyes, okay, or these imaginal discs here that are the imaginal disc of the wings that in the adult they will give rise to the wings, okay, and it's perfectly well known how it happens because it has been very, very well studied. Here I have two images of two videos, but they don't work. Doesn't matter. This was the development of zebrafish and a Drosophila for you to compare how different they are. Okay. Again, during development, something happens. Okay, that makes that you have an egg that in theory is round, okay, it doesn't have symmetry. The sperm enters in one side of the egg, and then this zygote begins to divide, okay, and then more and more and more, okay. How can it happen that at the end this mass of cells that, for example, so beautiful animals, no, and so symmetric animals. How can it happen? This happens because between cells there is a continuous communication. Continuous communication. Okay, this is very important. And for cells to communicate, what they use is the signaling pathways. Okay, this cell here, and this cell here, and this cell here. Each of them know their position, okay? And this position, they know because they receive signals from here, and maybe, or for sure, also from here, because here, surrounding the cells, there are things. There are extracellular matrix, there is yolk, there are many things that the cell sends, okay? This is how they know it. And for example, one of the things that happen during development is that from the entry point of the sperm in the opposite side, for example in Xenopus development, not always, okay, but in Xenopus development, in the opposite side it will accumulate some factors that are called the maternal factors. Maternal factors are mRNAs and proteins that are already in the egg, okay, and they are accumulated in a different way. For example, this is the animal pole and this is the vegetal pole, and they are accumulated in a different way, in one side or the other. And that's the reason why, for example, during the cleavage, this cell here will have this factor, but this cell will not have. So, and so on. Okay, the signals that are surrounding the cells give them the information. Another thing that can happen, some animals have a development that is regulative, okay? and others it's determinative. It means that, for example, at this stage, the animal that has a regulative development, if you cut in two parts, what will happen? What will happen? What do you think? Sorry? They will give rise to two embryos, two normal embryos, because they sense that something is missing and the cells are able to reorganize again. This is the regulative, okay? And the regulative development is until certain stage. For example, at when you already have 300 cells, then it's impossible, okay? It has a limit, yeah? And the determinative, what happens is this: if you divide this embryo in two, okay, or this way it's not so easy, but this way, for example, because this normally this becomes ectoderm, okay, and this becomes endoderm at the end of a normal development. What happens is that in the determinative development is that this part will form only endodermal tissue, and this part only ectodermal tissue, okay? But not a whole embryo, any of them. Okay? In this slide, what I wanted you to have the idea is that already from the egg, for example, the signals that establish the axis of the animals, okay, the anterior-posterior and the dorsoventral axis of the animal is already established, okay, because of the maternal signals. So then maybe nobody knows what is the Spearman organizer. This is a very common thing in developmental biology. You have never heard about it, no? I told you about it just for you to understand how it works in communication between cells. This is an embryo of Xenopus again, and in this part here, some proteins, like beta-catenin, for example, accumulate, okay? They accumulate here, and together with other signals that are in the ventral part of this blastula, they here organize what is called the Newcob center. Okay, the Newcob center is, if you have an embryo, okay, this embryo now has many cells, okay, many cells. It's at the blastulate stage. It's just before gastrulation, okay, just before the formation of the germinators. And here, it's formed what is called an organizing. An organizing center is some cells that express some proteins that are secreted to the extracellular medium, and then these cells here receive the signal, okay? In a way that these cells that are very near receive a lot of signal, and these cells that are far away do not receive the signal. And in this way, what do you get? From a ball of cells, now you have a polar, something that is polar, that it has polarity. Okay, this is what is an organizing center. Okay, it's cells that secrete molecules that are able to pattern, okay, to give different identities to the field that is next to them. Okay, for example, in the case of the formation of the limbs during development, in the limbs, okay, during development, the organizing center that is here will say, here you must have the arm, and here you must have the hand, and here the digits, okay? And this is because the organizing center says it. What happens if you, for example, put an ectopic organizing center here? That it will appear a hand, and here another. Okay, this is what will happen because it has organizing activity. Okay, and